Hello everyone, I am Dr. Sirajul Faisi. I welcome you in my YouTube channel. Today in this video, I am going to talk about enzyme. You know my friend, enzymes are a biological catalyst. It is an action of saliva on the conversion of starch into sugars and action of gastric juice on digestions of meat gave birth to the idea of the presence of catalyst in biological system. My friend, these biological catalysts were named as ferments on account of the fermentation of sugar into alcohol by yeast. First time these ferments named enzyme by Louis Pasteur in 1860 and Buchner 1897, who first extracted the enzyme from yeast, which could still induce fermentation of sugar. Now, properties of enzyme. First, enzymes are complex macromolecules with high molecular weight. Second, they catalyze biochemical reaction in a cell. They help in the breakdown of large molecules into smaller molecules or bring together two smaller molecules to form a larger molecules. Next, enzymes do not start a reaction. However, they help in accelerating it. About the enzyme affect the rate of biochemical reaction and not the direction of the reaction. Most of the enzymes have high turnover number. Turnover number of an enzyme is the number of molecules of substance that is acted upon by an enzyme per minute under saturated substrate concentration. High turnover number of enzyme increases the efficiency of the reaction. My friend, enzymes are specific in action. Enzymatic activity decreases with increase in temperature, and all enzymes show maximum activity at an optimum of 30 to 40 degrees Celsius. They show maximum activity at an optimum pH of 6 to 8. The velocity of enzyme increases with increase in substrate concentration and then ultimately reaches maximum velocity. Now the last point, one of the characteristic properties of enzyme is that they act over a specific substrate. The specificity may be broad, such as a group specificity, or absolute, such as optical specificity. Now, my friend, mechanism of enzyme action. For certain substances to undergo chemical reaction, they have to be provided with enough of free energy to attain an activated state called transition. And this energy of activation acts as energy barrier, and its absence, reaction fails to occur. Energy barrier is of different magnitude in different reaction. If energy barrier is low, the large number of molecules will be in activated state, which means that the reaction rate is fast. Conversely, if the energy barrier is higher, smaller number of molecules is in activated state, and thus the reaction is slower. Enzyme is active in catalytic action of biochemical reaction. They act on substrate and forms a complex after interaction with the enzyme is called active center. The enzyme and substrate forms a complex at active center. Now, this binding action makes both enzyme and substrate stable. The interaction between substrate and enzyme may be either ionic bonds and hydrogen bond or van der Waals forces. The active site of enzyme have some special groups such as NH2, COOH, SH, which bind the substrate through above bond to form a transitional intermediate compound called enzyme substrate complex. Look at this picture. Without enzyme and with enzyme reaction is fast, without enzyme reaction is slow. Now, type of mechanism of enzymes. There are two types of mechanism involved to explain substrate-enzyme complex formation. First, lock and key theory. In 1894, Emil Fischer explained the specific action of an enzyme with a single substrate using a theory of lock and key analog. According to this theory, reaction of substrate and enzyme is analogous to lock and key. Enzyme is analogous to key, where the geometrical conformation of socket is fixed. Similarly, substrate has also got fixed geometrical configuration like that of key. A particular lock can be opened or closed by a particular key. According to the particular substrate can be found at active site of particular enzyme forming substrate-enzyme complex. My friend, enzyme substrate complex remain in tight fitting and active site of enzyme are complementary to substrate molecules. Subsequently, enzyme substrate complexes result in the transformation of substrate into the product formation due to activity of reaction site. Since product has lower free energy, it is released, enzyme are fixed to receive another molecule of substrate and thus enzyme activity continue. In this analogy, the lock is the substrate and the key is the enzyme. Only the current size a key fit into the keyhole, active site of the lock enzyme. Smaller keys, larger keys, or incorrectly positioned teeth on keys incorrectly halved or sized substrate molecule do not fit into the lock enzymes. So look at this demonstration. This is active site, enzyme substrate complex. Now, induced fit theory. In 1958, Bushland modified the Fischer model for the formation of an enzyme substrate complex to explain the enzyme property more efficiently. According to Fischer model, the nature of the active site of enzyme is rigid, but it is able to be pre-shaped to fit the substrate. Bushland explained that enzyme molecule does not retain its original shape and structure, but the contact of the substrate induces some geometrical changes in the active site of enzyme molecule. The enzyme molecule is made to fit completely the configuration and active center of the substrate. At the same time, other amino group acid residue may become buried in the interior of the molecule. This theory can be explained by the hypothetical illustration. You can see here hydrophobic group, the conformation change brought about by induced fit in an enzyme molecule. The hydrophobic and charged group both are involved in substrate binding. The phosphoserine SH groups of cysteine residue are involved in the catalyst. Residue of the other amino acids such as lysine and methionine are not involved in either binding or catalyst. In the absence of substrate, the substrate binding group and catalytic groups are far apart from each other. But the contact of the substrate induces a conformation change in enzyme molecules and align both the group of substrate binding and catalysis. Simultaneously, the spatial orientation of the other region also change, and this causes the lysine and methionine much closer. So look at this substrate enzyme active site of enzyme. Enzyme change shape slightly as substrate bind, and this is the product. So my friends, I hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.